Know that this is the best time of the year, and if you had come last weekend you would have had 85-degree weather for 4 straight days. That was our summer, and now we're into fall. So I'm going to talk about sedation and natural sleep, and this has come up a few times. I have a slightly different take on things, but I don't think it's too controversial. I want to speak about the effect on cognitive function, and it should become clear during the talk why I think this is an area that we really have to consider sleep disorders as a very important cause of morbidity and mortality, perioperative or otherwise. So the questions that I'm going to address are, how does sleep enhance cognition? Because we do know that it does that. What effect do these sedative hypnotics have on cognitive function? And I'm going to concentrate again in the patients in the ICU. I think the patients in the ICU are a real problem, as Mark has alluded to. And the other group are the neonates. It turns out that if you're a preemie, the average time that you spend on a ventilator in the ICU is 38 days. So that's a lot of time in the ICU, and it's a lot of sleepless nights at a very vulnerable time. But I'm going to talk about the adult ICU. Then I'm going to talk about whether sedative hypnotics actually converge on the mechanisms that produce sleep. So I do have a potential conflict. It is potential, I'm afraid. I patented dexmedetomidine for its use in 1987 with Mika Shainin. We reassigned our rights to the patent to a company called Pharmos that synthesized the molecule. It was developed by Abbott Labs and Aspira. They gave me, at the time that I was at Stanford, $250,000 went into my lab over 5 years. It's now selling $1 billion a year. So that wasn't a very good investment. And I accrue no financial benefit from this. So the first question is, how does sleep restore cognitive processing? So first I'm going to show you some data, both from ourselves, from Souvakis and another lab, about what sleep deprivation does to cognition. And for this, we use the trace sphere conditioning apparatus. So what you do is you put the rodent into this chamber, and you, after a certain amount of time, you give a tone, and then a certain time later you give a shock. And so when this animal is put back into the same context sometime later after training, they remember that they were in this environment in which they received the shock, so they assume freezing behavior. And that freezing behavior is a manifestation of remembering that they've been in that context before. So a decline in freezing behavior means their memory has declined. And so this is work from G's lab from Boston. And here he is deprived sleep for 24 hours. And he shows that the amount of freezing behavior compared to the control or the well-rested animals, their freezing time significantly decreases. Work from Souvakis, looking at sleep fragmentation. Here we're doing an orbital shaker, not exactly the same as Mark's apparatus, but it's an orbital shaker. The rats were not monitored by EEG, so we can't definitively say they were what phase of sleep they were in, but they were observed throughout. And so if you do the novel object recognition test, if you put a mouse into an arena where they see the same object or a novel object, they will spend more time with the novel object than with the familiar object. However, after sleep deprivation, they don't remember that they've seen that object before, and so they spend the same amount of time with both the novel and the familiar object. So this is evidence that there is a problem with memory. Now, we do know that memory is enhanced during sleep, and there's 2 dominant theories about why there's this memory enhancement during sleep. The first is that there is some reorganization of the signal that originates in the hippocampus that is transferred to neocortical stores. This is a sort of a reorganization, and this is called the systems level memory consolidation. This is the term memory consolidation, and it's one form of memory. The second is the synaptic homeostasis hypothesis, in which there's a change in the synaptic efficacy so that the signal to noise ratio goes up. And this is definitely driven by process S, the homeostatic process of sleep. It accumulates during wakefulness and discharges during sleep. And the work really comes out of Giulio Tononi's lab with Maria Chiarelli, and I'll show you. This is a cartoon that's in Scientific American May edition, I believe, showing this work. Now, so what you're looking at here during the awake state, when you're learning, you're forming these synaptic connections over here. This is synaptic plasticity, sort of memory, but you're also forming some irrelevant memories as well over here. You're forming some connections. They're not really things that you really want to remember. And some, during sleep, you reinforce the ones that you need to remember in the way that I've just showed you with this reorganization, and you eliminate the other ones, the ones that you don't need. And this is quite remarkable. How does this happen? So just to remind you of what is synaptic homeostasis. So this is a driver to sleep that increases during wakefulness. And in the case of this theory, it's due to synaptic potentiation. And then there is, during slow wave sleep, there is a synaptic downscaling that occurs. And it's shown graphically over here in this cartoon. So during wakefulness, you have a lot of learning going on, and so a lot of synaptic potentiation occurring. And this is what happens with long-term potentiation. And then during slow wave sleep, you have the synaptic downscaling so that these irrelevant ones that you don't need any longer disappear, and the synapse is downscaled, if you will. And it only occurs in slow wave sleep. And the reason why you need this is because it's like a data bank. If you didn't get rid of the memories that you no longer need, if you didn't extinguish them, then you just would run out of space and energy. And so it's a space problem. And this is a way of getting rid of the stuff that you don't need. Now, how you filter between the stuff you need and the stuff you don't need is not clear to anybody. But it is quite remarkable. Now, during wakefulness, for example, you see an increase in the spines on the dendrites. If you stimulate the neurons, you see a much bigger amplitude of response. And also, you heard about AMPA signals. There is an increase in AMPA expression at the synapse during wakefulness, which disappears during sleep. And the dendrites disappear during sleep, and change in the shape of the curve after stimulation also occurs. Now, so Tononi's lab wanted to find out why this happened, and they looked at the changes between gene expression from wakefulness to sleep, and there was a very prominent increase in brain-derived neurotrophic factor, BDNF, as you can see in the black bar here. So the difference between wakefulness and sleep is in the very marked increase in BDNF. Now, BDNF has a very many activities, but importantly, it is one of the drivers of synaptic plasticity. It's also an important neuroprotectant as well. So during wakefulness, you get accumulation of BDNF. So the question they asked was this the driver of that induced slow wave sleep? And so what you're looking at, first of all, is that wakefulness-induced BDNF over here is positively correlated with the subsequent duration of slow wave sleep activity, which is over here. So here's a nice strong correlation with the amount of BDNF that accumulates and the subsequent amount of sleep that happens. Well, that's just an association, it's a correlation. But here, they've exogenously administered BDNF, and found an increase in non-REM sleep as well as an increase in slow-wave sleep activity with infusion of BDNF. And when you do the converse, where you give an antibody to BDNF, you see a decrease in non-REM sleep and a decrease in slow-wave sleep activity. So this seems to be a fairly good demonstration that BDNF is quite important in driving this sleep, and I'll just recap the findings. So sleep deprivation definitely impairs cognition. And by the way, Mark, both the groups, the She group and Sue, also found some neuroinflammation in this 24-hour period. The slow-wave sleep activity downscales synaptic potentiation. As I say, it's probably the space issue. That BDNF accumulates during wakefulness and drives the slow-wave sleep activities I showed you, and also downscaling, and that BDNF slow-wave activity and downscaling are required for normal cognitive processing. So next, let's just consider the issue of the sedative hypnotics in the ICU and what happens to cognitive function in these patients. Mark showed some nice hypnograms. I think Sleepless in Seattle, right? Well, this is Sleepless in the ICU. Very long periods of wakefulness. Deep sleep is almost never found. And, well, REM sleep is virtually non-existent, as you heard. And total loss of the circadian rhythm. So I don't have to explain that to you. But here's some hypnograms of several patients. You can see they never get into the deeper stages. Look at this person over here. And here's a person sleeping day and night, and this person's hardly sleeping at all. So you can see it's really problematic in the ICU with patients and their sleep. So what are the problems? The problems are that we use prolonged sedation in these patients, and we have problems with weaning from mechanical ventilation. They stay in the ICU a long time. It's very difficult to do a neurologic examination on these patients, especially when they're deeply sedated. And also deep sedation, or rather deep sedation with some drugs, definitely predisposes to infection and to delirium. So enter two colleagues of mine, Wes Ely and Pratik Pandey. They are both intensivists from Vanderbilt, who we teamed up with a few years ago to study the effects of dexmedetomidine versus a benzodiazepine for sleep, for providing sedation, looking at the effect on cognitive function, i.e. delirium, postoperatively. So this trial was randomizing either MICU-SICU patients to lorazepam, which is still the most common benzodiazepine used in the ICU, or dexmedetomidine. And they were supplemented with fentanyl in both cases. And you'll notice that the amount of delirium and coma-free days is significantly greater in patients who receive dexmedetomidine. And we also found a couple of other things that was quite interesting. Now, the mortality rate, very small study, and it wasn't powered for mortality, that there seemed to be a benefit, a mortality benefit conveyed by dexmedetomidine. And those of you who are going to come to the neurobiology of sleep, or no, neurobiology of, what's the title of our panel?
I think it's Interfaces of Sleep and Anesthesia.
Interfaces of Sleep and Anesthesia on Monday. Okay. Sometime during the ASA. Okay. Well, I will speak more about that. We're going to speak about both. Ken and I are going to speak about this problem as well. But I'm going to talk about the immune system a little bit more, and try and understand whether the mortality could be due to the immune system. If we just took the patients who were septic at the time of admission, this is the mortality rate in those who randomized to receive dexmedetomidine, and the mortality rate in those who received lorazepam. And there was a hazard ratio of 0.3, i.e. a 70% reduction in expected mortality. Now, this study was not powered for this endpoint, and we didn't enter septic patients. We did a post hoc analysis. So just remember that when you interpret these data. So Riker repeated the study that I just showed you. He did dexmedetomidine versus midazolam, and also supplemented with fentanyl, and basically showed pretty much the same thing, that there was a significant decrease in delirium in the patients who received dexmedetomidine. The duration of delirium was decreased, time to extubation was decreased, length of stay in the ICU was decreased, and also found the infection rate was decreased as well. So that's the study by Riker. Now, so delirium. When I grew up, when I went to medical school, we used to talk about sundowning, okay? And this is something that happened in the afternoon, the late afternoon, and the patients eventually got better when they left the ICU. It also was called ICU psychosis, and it just, you had it in the ICU, left the ICU, you're over this. No, you're not over this, and this is a very nice study that is in the New England Journal of Medicine last week, looking at global cognition scores after you get delirium in the ICU. And we're talking about 3 months, 12 months later, and this is in the under 65s, the over 65s. Doesn't seem like young age protects you against this. You get severe enough cognitive decline, long-term cognitive decline, of the equivalence of TBI, and early Alzheimer's. So this is a big concern. If you get delirium in the ICU, this is a problem. And it's a big problem with duration of delirium. So the longer the days of delirium you have, the more likely you are to have a lower score and come into those levels where you're TBI and Alzheimer's. So delirium in the ICU is a huge problem. Anything that decreases delirium in the ICU, including improving sleep, is something that should be desirable. In fact, if you look at the dose of lorazepam, and of course these are drugs of addiction, so you need more and more each day in order to get the same effect. And once you get to about 24 milligrams, 100% of patients, 100% will get delirium, will become delirious in the ICU. So this is definitely a drug. If you want to study delirium, give patients benzodiazepines. If you don't want to study delirium, avoid benzodiazepines. Okay, so the big question, I guess, that everybody's been dancing around is, how close are sleep mechanisms, or how close are sedatives to sleep? And I don't think we really understand that, but I'll give you my view on this. We've heard Cliff Saper's name mentioned over and over. Cliff and I teamed up some years ago to study this, looking at markers of neuronal activation. And this is his classic description of the arousal system in the brain, consisting of noradrenergic, cholinergic, orexinergic, and histaminergic fibers. So, and they go ultimately to the cortical, subcortical regions, where they keep you awake in a way that is quite difficult to understand, but in rodents at least it's been well identified. Now, you'll notice the important role of a couple of nuclei here. One is the locus ceruleus, the other is the tuberomammillary nucleus, and the preoptic nucleus as well. So I've just simplified this, although it doesn't look that simple to me looking at it now. The difference between the activities in these nuclei in the awake state and the non-REM sleep state. So you'll notice that in the awake state, VLPO, ventrolateral preoptic nucleus, is switched off and switched on in non-REM sleep. Conversely, LC, dorsal raphe, perifunicular, tuberomammillary are all active, releasing these amines and so forth that cause the arousal to occur during wakefulness, whereas in non-REM sleep you don't, you see these switched off, whereas the Vilpo is switched on. So that's work that we did, this is work that Cliff Saper did. So we looked at activity in various nuclei during, and what we found surprisingly is when you use a GABAergic drug, you get immobilization, but you don't get the same kinds of changes that you see with natural sleep. And the big change is that the locus coeruleus doesn't switch off. The locus coeruleus remains active. There is an increase in Vilpo and a decrease in tuberomammillary nucleus. Now, in this study we did in London when I was at Imperial College, looking at a single subject, and we did it, only caught a few subjects during an fMRI, and we're looking at the bold signal under two conditions. One is the subject's natural sleep and the subject's sleep induced with midazolam. And we're looking at a subtraction scan. So these voxels are activity that occurs in subjects where there's a difference between the sleep, their natural sleep, and the sleep that is induced with midazolam. So those in the audience who are anesthesiologists and say, I'm going to give you something that's going to make you go off to sleep now, nothing can be further from the truth. This is not sleep. Benzodiazepines do not produce sleep because this picture is very different from the subject's natural sleep. And we caught two subjects in this, and Sue Backus is now doing a study with magnetoencephalography, looking at the difference, with Phil Curian as well, looking at the difference between subjects getting propofol and dexmedetomidine to see if we can see differences. This is the EEG with subjects that receive diazepam. You can see a decrease in non-REM sleep. Now, cognitive function. This is, we're looking at the improvement in cognitive performance with different drugs. This is, the drug here is midazolam. So this is the change. So as you have more sleep with this drug, your cognitive performance actually goes down, which is the converse of everything else that happens with sleep. So although we get chemical immobilization with benzodiazepines, we certainly do interfere with cognitive performance. And guess what? When you give diazepam, for example, you get a decrease in brain-derived neurotrophic factor. And this decrease is due to its activity at the GABA receptor, because if you eliminate one of the subtypes of the GABA receptor, one of the subunits of the GABA A receptor, you don't get this effect. So benzodiazepines do not cause sleep. They cause a state they're immobilized, and they have an EEG picture that's different from sleep. They have a profile with respect to their neurotrophins that it's quite different from sleep. So enter alpha-2 agonists. We've been working on these for the last, it's 30 years now. This is clonidine, which is a non-selective drug. Almost equal activity of both alpha-2 and alpha-1, or rather about 200 to 1. Dex is 1600 to 1. It's got better pharmacokinetics, and so we've done most of our work with dexmedetomidine. And this is nice work by Tom Ebert, who had the courage to give volunteers, get them up to a concentration of 14.7 nanograms per ml. Now that is 20 times the therapeutic levels that are used in the ICU. There is no other drug in anesthesia that we use that we can give 20 times the dose of and not have to support either ventilation or the cardiovascular system. But in fact, he didn't have to do either in these subjects and produced a nice dose-dependent sedative effect in these individuals. We worked out the mechanism, the molecular mechanism. It works at an alpha-2 adrenergic receptor. Of course, it's an alpha-2 agonist. You'd expect that. Through a couple of species of G proteins, they couple to ion channels. One prevents calcium from coming in, the other facilitates potassium from going out, and the net effect is membrane hyperpolarization. This is happening in the locus coeruleus, and the subtype is an alpha-2A. This results in a decrease in the firing of the locus coeruleus neurons, which we believe is important for perhaps initiating this response. So if we could just show you what happens in non-REM sleep, the changes that we saw with dexmedetomidine were very similar. Activation of VLPO, and these are the nuclei, LC, perifunicular nucleus actually does increase. It's the one thing that's different is the perifunicular nucleus. So if you want to just think about dexmedetomidine, it decreases firing LC, increases VLPO activity, decreases the activity of these other neurons, and presto, you go off to sleep. So if you look at the difference between the bold signal there were two subjects, so this is from one subject, the difference between the natural sleep and the sleep induced with dexmedetomidine, you can see that these are quite different. They're quite similar. There are not many voxels of activity. That means that they were quite similar, this subtraction scan. And you do see something that looks vaguely like a sleep with sleep spindles, but it's not exactly like a slow wave sleep. And I think, but it has some resemblance to sleep, let's put it that way. Now, this is work that was done in vitro by Vincent Degas, who just published this work in Anesthesiology this year, showing you that DEX does increase the amount of BDNF, and this is happening through an alpha-2 adrenoceptor because he could eliminate this effect with yohimbine. So dexmedetomidine, unlike the benzodiazepines, it does increase BDNF, whereas benzodiazepines decrease it. So again, difference is locus coeruleus is active in the GABA, perifunicular nucleus is active, whereas locus coeruleus is inactive, perifunicular nucleus is inactive in GABA, it's active during sedation with the alpha-2. Locus coeruleus is active in GABAergic agents and inactive in alpha-2s, and that seems to be a difference between those two classes of drugs. So our interpretation is that the hypnotics do converge in sleep pathways, but not at the same point. Alpha-2s seem to converge at the level of the brainstem. Benzos seem to converge at the level of the hypothalamus, and they result in qualitatively different hypnotic response. The alpha-2s produce a slow wave sleep activity, whereas benzos, that's a term that I've started to use, causes chemical immobilization. So let's just think about what happens with sedatives in the ICU. The benzos are likely to cause what Wes Ely calls acute brain failure. This hypnotic effect doesn't really have the restorative properties, cognitive restorative properties of slow wave sleep activity, and later I'll show you about the effect on infection and sepsis. Whereas dexmedetomidine seems to induce something that resembles sleep and probably facilitates the restorative properties of sleep on cognitive function, and we have some data to show that it maintains the innate immune response. So I'm going to conclude by saying that sleep deprivation clearly does cause cognitive decline, that BDNF is required for this, for the slow wave activity and the synaptic downscaling that is necessary during normal sleep to increase that signal to noise ratio that is necessary for improvement in memories, that cognitive decline occurs if synaptic downscaling does not occur, and that cognitive decline occurs with prolonged sedation with BDZs, but less with alpha-2 agonists, and that alpha-2 agonists, but not BDZs, cause an increase in both BDNF as well as slow wave sleep activity. So what other things are we interested in? Well, first of all, whether the difference between the alpha-2s and the GABAergics on synaptic downscaling, don't know that. Immune function, I'll report that on Sunday. Connectivity using MEG is the study that Sue's doing right now. With Wes Ely and Pratik, we've just got funding from the NIH to do a controlled trial, randomized controlled trial comparing Dex versus propofol for sedation in mechanically ventilated septic patients. We are proposing to correlate that with sleep architecture so that we can see whether, in fact, that's going to be an improvement. I'll just point out that in the one study where propofol anesthesia and BDNF was looked at, there was a significant decrease in this is circulating BDNF after induction of anesthesia with propofol. This is from Lajlo-Butskits. And finally, what about the effects of sleep deprivation in the postoperative patient? We have been investigating postoperative cognitive decline, and Sue's now looking at the combination of what happens when you have surgery, cognitive decline, and then you superimpose on sleep deprivation to see if we have a sort of a double hit. So I'll finish there and thank those people that did the work and the people that supported this. Thanks very much.
We have time for a few questions. Atul?
That was great. Two questions, if I may. Both
Yeah.
of which may be naive. There's some evidence, I think, in some clinical experience that suggests benzodiazepine infusions may be very different from intermittent dosing of benzodiazepines. Dave, you showed compared to dexmedetomidine, compared to infusions rather than intermittent dosing.
They were infusions, that's correct, yeah.
Do you have any reason to believe that, I mean, intermittent dosing may just limit the dose of it, so you may just be dosing it more cautiously or more prudently. Was there any other reason, the mechanisms that Rich Horner was talking about in terms of changes in GABA receptors, are those likely to be different?
You think this is a kinetic issue then?
I'm wondering.
Well, I don't know that in the ICU it's invariably by an infusion. So I
We
don't.
don't do that anymore. We give it intermittently because,
You do it intermittently now.
yeah, based on requirements of the patient.
Okay.
And that,
Okay. Well,
Maybe
that's
it's
an
safer.
interesting. And are you? I mean, are you looking for the effect on delirium in these intermittent administrations?
yeah,
And are you seeing it less?
I mean, it's possibly
Okay.
premature to say that, but the existing data, I think, support intermittent dosing. That's being done more, but it may just be a more prudent way of providing the drug rather
Right.
than sedating the hell out of everybody.
Right. I think benzos are really going out of favor in the ICU.
Yeah.
I hope I've had something to do with that. But yeah, that's a good. What was the second one, Atul?
The other question related to BDNF, so I think Gordon Mitchell and these guys have been studying neuroplasticity and
Yeah.
facilitation of BDNF. Do you have any reason to believe that pharmacological induction of BDNF would have result in improvements in
Sleep. Well,
neuroplasticity and LTF and these things that they measure, so
yeah.
memory and other aspects of neural.
Yeah, so Tononi is, so we're all awake, hopefully, right? Okay, and so right now, and you're learning something, hopefully, right? And so you're just pushing up your BDNF, and you're going to sleep really well tonight because you're just got a lot of BDNF that you're accumulating. But so you're having two things happening. You're having synaptic potentiation, and you're accumulating BDNF. And the synaptic potentiation is probably driven by the BDNF. So I think they're so inextricably linked, but you have to have the BDNF during the wakefulness to get the sleep that follows. That is the homeostatic driver. So did I answer your question? I don't think I did.
I think my question was vague because I'm naive to this topic, but my understanding is that you knock out BDNF, that they don't get long-term facilitation.
No, that's correct.
The pharmacological drugs that induce BDNF, is it likely to be beneficial, or is there some?
I think beneficial. I mean, I hope I suggested to you that benzos push it down. Propofol seems to decrease it, whereas Dex seems to increase it. I think BDNF may well be a driver. to sleep in some way. That's the Tononi model.
One
Yeah.
other quick question.
Do you have any thoughts or information regarding the use or the effect of other GABA agents like sodium oxybate or
GABA B.
GABA B?
Do you mean the sort of date rape drug? Is that?
Well, it's used for, it induces slow wave activity. So we use it in narcolepsy and in some patients, the amount of slow wave you see is incredibly robust. And baclofen has also been, there's controversy, conflicting data on that, but as an oral agent also seems to induce slow wave activity in contradistinction to the GABA A agonist.
Right.
So I'm interested in any thoughts you might have experience with GABA B agonists.
We
They
don't.
have different
We've-
effects.
No, we've never investigated GABA B agonists.
I can tell you that in subjects that we've looked at who've been treated with Xyrem or sodium oxybate, that the slow wave pattern that you see on the sleep architecture is incredible. I mean,
Oh.
I've got one case, it's 97% of his sleep time is slow wave activity.
Is it definitely GABA through a GABA B receptor though?
It's GABA B.
I mean, have you given a GABA B antagonist and eliminated that effect?
Oh, I have no idea about that.
Okay. What happened to Ken?
He's
Oh,
behind.
there you go. Okay.
So now it's a pleasure to introduce Ken Saul, again from Harvard and Mass. Who'll be talking to us about the other end of the spectrum, not induction, but emergence.
Thank you very much. I'd also like to thank the organizers for inviting me to speak to you. And as Mark suggested, I'm going to be switching gears a little bit from sleep circuits, transitioning over to arousal circuits and the work that we've been doing on what we've been calling reanimation from general anesthesia, specifically focusing on the role of dopamine in arousal and emergence from general anesthesia. So in current clinical practice in anesthesiology, when we want to wake up our patients in the operating room, it's a completely passive process. What we basically do is we shut off the general anesthetic and we allow the pharmacokinetics of anesthetic drug clearance dictate when the patient will wake up from general anesthesia. And as any anesthesiologist here can tell you, there are a lot of clinical problems associated with emergence from general anesthesia. There was a retrospective review of over 80,000 anesthetics that was published in 2002 that found that after problems with intubation, the second biggest source of problems, severe intraoperative problems, actually occurred during emergence from general anesthesia. And most of those problems were due to what they found were misjudgment of residual anesthetic effects. Emergence delirium is a common problem, especially in pediatrics. Any pediatric anesthesiologist will tell you that this is a major problem. The incidence has been reported in the literature to be as high as 30%. And there was an interesting study published in 2004 that found that having the parents around when the child wakes up from anesthesia actually didn't change the incidence at all, suggesting that it's not an environmental issue, it's really a pharmacological issue related to the general anesthetics. There was an interesting paper published last year in the New England Journal that found that in patients who had cardiac surgery, the ones that had emergence delirium had a higher incidence of cognitive impairment even up to a year after the anesthesia, suggesting that the neural circuits involved in emergence from general anesthesia may actually be also involved in restoration of cognitive function as well. And finally, delayed emergence is a problem that we deal with all the time in the operating room. This is something that can be quite costly if you consider that OR time can cost anywhere upwards of $1,000 an hour. Even a 10 or 15-minute delay in wake up from the time that the surgery ends to the time when the patient actually wakes up and gets extubated and leaves the operating room is essentially dead time in the operating room. Nothing happens. The room can't get turned over. Everyone's just kind of sitting around and waiting. And if we could have some ways of optimizing that and actually playing some role, an active role in having our patients wake up from anesthesia, this may actually improve efficiency in the operating room as well. And so our research group has been interested in what we've been calling reanimation, which is basically this concept of taking a subject in an anesthetized state, performing some sort of an intervention, and actually sort of waking them up, pulling them out of that anesthetized state back to the awake state. And so our approach is something, it's a little different from drug reversal historically in anesthesia practice. We anesthesiologists reverse drugs in the operating room all the time. We routinely reverse neuromuscular blockade, at least when we have the drugs available to do so. Right now we have some drug shortages that are causing some problems there. But in general, we do have ways to reverse all these drugs. We have ways to reverse opioids, we have naloxone, we have benzodiazepine antagonists like flumazenil. We can also reverse heparin with protamine. We reverse warfarin with vitamin K, FFP. So we're used to this concept of reversing drugs in the operating room. We do it all the time. But the problem is that we know the molecular mechanisms of these drugs. And so the strategy historically has been to create molecular level antagonists to block the effects of these drugs or to somehow cleverly circumvent them or to reverse them. For example, neuromuscular blockade, we use, we sort of manipulate the cholinergic pharmacology to reverse the actions of these drugs. But the problem is that for general anesthetics, the molecular mechanisms are much more complicated and they're far less cleaner than they are for drugs like this. And so historically, developing molecular level antagonists for general anesthetics has been really an unrealistic approach for reversing these drugs. But we took a different strategy, a different approach, and we hypothesized that general anesthesia may actually be reversible not at a molecular level, but rather at the level of neural circuits. And some of those circuits have actually been discussed in the talks today. This is a diagram of some of the arousal-promoting circuits, similar to the diagram from Cliff Saper's review paper that Mervyn showed in his talk. And I just wanted to kind of emphasize that there are quite a few arousal-promoting nuclei in the brain, and there are quite a few different neurotransmitters that are involved in arousal. There are the cholinergic circuits in the pedunculopontine tegmentum, lateral-dorsal tegmentum, and the basal forebrain. There are also monoaminergic centers, norepinephrine, dopamine, serotonin, histamine. You've heard about some of these earlier today. These monoaminergic neurotransmitters are also involved in promoting arousal. And orexin is also, it's a peptide neurotransmitter that you've also been hearing about today as well. And we think that the preoptic area, which releases ghrelin and GABA, is the primary sleep-promoting center in the brain. And so there was a paper published by Langzhou and Michael Alkire and their colleagues last year in J Neuroscience, which is quite compelling. This was a PET scanning study in patients that were essentially waking up from anesthesia. And what they were basically looking for what part of the brain is actually becoming activated when patients emerge from general anesthesia. They did propofol anesthesia, let the propofol wear off, and looked at the parts of the brain that lit up when the patients first woke up and opened their eyes. And they also did this under dexmedetomidine sedation/anesthesia, where the concentration of dexmedetomidine was kept the same and they gently aroused the patient. And so it wasn't really a pharmacokinetic issue in that case like it was for propofol. And in both cases, the same parts of the brain lit up. And the parts of the brain that lit up were actually the parts of the brain that I just talked to you about. It was the upper brainstem, midbrain, thalamus, and a small part of the anterior cingulate cortex that seemed to consistently sort of turn on when patients were waking up from anesthesia, suggesting that it's these sort of phylogenetically conserved, sort of primitive parts of the brain that we share in common with rats and other animals that may be the sort of the principal sites that get turned on when we emerge from general anesthesia. So we became interested in this concept of, can we activate these arousal centers in the brain using drugs that are already available and are already being used in humans now? The neurotransmitters that we were particularly interested in were dopamine and norepinephrine, because we knew that methylphenidate, more commonly known as Ritalin, which is a very commonly used drug to treat attention deficit hyperactivity disorder, acts by inhibiting dopamine and norepinephrine reuptake transporters in the brain. So if this is a cartoon of a dopaminergic or noradrenergic synapse, the neurotransmitter is in red, it's getting released into the synapse, it does its thing, and then there's this reuptake transporter that recycles it back to the presynaptic neuron. And what methylphenidate does is it blocks that reuptake transporter for both norepinephrine and for dopamine, thereby raising the levels of these two arousal-promoting neurotransmitters in the brain. So we hypothesized that methylphenidate may actually be efficacious for reversing general anesthesia or promoting arousal in anesthetized animals. And just to summarize some of our previously published work, we did indeed find that methylphenidate induces what we've been calling reanimation, or an active emergence process. We really want to differentiate this from passive emergence, which is when we basically just let the drugs wear off, because the mechanisms may actually be different underlying that kind of wake up versus what we're doing, which is to sort of activate these arousal pathways and stimulate wake up. And we also found that methylphenidate induces reanimation from propofol anesthesia. And this, to us, was pretty good evidence that this is probably a circuit-level phenomenon. It's very unlikely that isoflurane and propofol are acting at exactly the same molecular site and that methylphenidate is actually blocking that and knocking it off. It's probably a circuit-level phenomenon like I described to you earlier. So our more recent work has been focused on other ADHD promoting drugs, other ADHD treatment drugs, and here are a couple of them that we've been looking at. One is called atomoxetine. It's marketed as Strattera. It's recently approved for treatment of ADHD. It's different from methylphenidate in that it's far more selective for the norepinephrine transporter than it is for dopamine. In fact, it's 1,000 times more selective for norepinephrine. So it's essentially a selective norepinephrine transporter blocker. And so it selectively increases norepinephrine levels in the brain. And then there's dextroamphetamine, which is similar to methylphenidate in that it increases both dopamine and norepinephrine levels in the brain, but differs in that it actually acts by an intraneuronal mechanism. It actually gets inside the presynaptic neuron and promotes release of dopamine and norepinephrine as opposed to blocking reuptake like methylphenidate does. And so we were looking at these two different drugs for ADHD, and this time we used sevoflurane, yet a different general anesthetic, just to sort of emphasize that this works for many different general anesthetics. The experimental protocol is quite simple. What we basically did was we kept rats in a sealed chamber and had them continuously inhaling sevoflurane at a dose sufficient to lose the righting reflex. And for those of you who aren't familiar with that, the righting reflex is a very commonly used experimental endpoint in rodents as a surrogate for loss of consciousness. And so when you take a rat and you put the rat on its back, it immediately wants to flip onto all fours. It's a very vulnerable position for a rat to be on its back. And so when the rat no longer flips over and stays on its back, we consider that loss of consciousness. And it turns out that the dose of inhaled anesthetics that produces loss of righting in a rat is comparable to the dose that causes loss of consciousness in humans. And so what we do is we establish a dose of sevoflurane sufficient to induce this loss of righting or unconsciousness in the rat. And then we give a dose of, normal saline through an IV. This is a control experiment just to ensure that the injection of normal saline through the IV is insufficient to induce an arousal response. And then 5 minutes later, we give either atomoxetine or dextroamphetamine. And what we found was that even at the highest dose of atomoxetine, there was absolutely no arousal response that we could find. The rats did not write, they did not flip back over, they basically stayed anesthetized, suggesting that noradrenergic stimulation is probably insufficient to reverse this depth of general anesthesia with sevoflurane. However, dextroamphetamine had a very dramatic arousal effect. It was dose dependent. At doses of 1 milligram per kilo and higher, we found that all of the rats had an arousal response and they all had restoration of this righting reflex. When we pre-treated the animals, not with normal saline, but with the D1 dopamine receptor antagonist, what we found was that we completely blocked this effect of dextroamphetamine and the rats no longer woke up, suggesting that dextroamphetamine-induced arousal is probably mediated through a dopaminergic pathway. And that's also confirmed by the fact that a noradrenergic stimulant, atomoxetine, failed to induce arousal. But when I talk about rats like flipping over, it's kind of hard to sort of visualize that as really an arousal response. They kind of flip or they don't. So I kind of wanted to show you a short video of what I'm talking about. So these are two rats that are continuously inhaling 1.6% sevoflurane. So we're talking about a submax dose, but a dose sufficient to induce loss of consciousness. As you can see, they're both flat out on their back. They're in these sealed chambers. They're both inhaling sevoflurane continuously. This animal, the one closer to you, just received dextroamphetamine. The one in the back did not, and now they're both receiving a saline flush. And so I want you to watch this rat here compared to his friend over here, and just remember, they're both inhaling the same dose of sevoflurane, but this is the one that received dextroamphetamine. As you can see, within a matter of seconds to minute, I mean, this rat wakes up and doesn't just flip over, but actually has behaviors consistent with arousal. And as you can see, this animal looks wide awake, even though he's still inhaling 1.6% sevoflurane, which is like a submax dose, which is pretty impressive, I think. As you can see here, this rat, that saline did not wake him up. So it's not the saline, it's the dextroamphetamine. We also did some EEG studies. These are spectrograms of EEG recordings from a separate group of rats. And just to orient you here, in these spectrograms, time is on the x-axis. I think this thing's kind of dying, but time is on the x-axis, frequency of the EEG is on the y-axis, and the power at any given frequency at any given time, which is the power is basically proportional to the square of the amplitude of the EEG signal. The power is denoted in color, so that high power, or having a lot of amplitude, a lot of activity at that frequency, is denoted in red. And when there isn't a lot of power at that frequency, it's denoted in blue. And as you can see, under sevoflurane anesthesia, there's predominant delta activity. So delta is like 4 hertz and below. What we find is that there's a lot of power at these low frequencies. So there's a lot of slow wave activity, essentially. And it's the same for this rat here. However, when normal saline is administered, nothing really happens. You wouldn't expect that to change. They're still having essentially this anesthetized EEG pattern. But after dextroamphetamine and atomoxetine, surprisingly, both of these EEG patterns rapidly change and go from a delta dominant rhythm to a theta dominant rhythm. Theta is about 4 to 8 hertz. And it was kind of interesting that even though atomoxetine didn't produce any behavioral arousal, it still produced a pretty dramatic EEG response suggestive of arousal. And so what this tells me is that perhaps the EEG change is driven more by the norepinephrine in the brain. So clearly norepinephrine, something's happening here. It wasn't that the dose was too low. This dose of atomoxetine was sufficient to induce a pretty dramatic EEG change. However, that EEG change is probably driven by norepinephrine, but it isn't sufficient to produce a behavioral arousal response. And so I think that when we look at these kind of papers, we really need to be careful and not assume that just because the EEG changed, that is then correlated with a behavioral change. Because here we had a clear difference between the behavioral experiments and the EEG experiments. And so I think that the data that I've showed you until now hopefully gives you a pretty good sense that it's probably dopamine that's driving the behavioral arousal response. So the next question that we asked ourselves is, so where is this dopamine coming from? When we give drugs like dextroamphetamine and methylphenidate, we're not actually giving an exogenous source of dopamine. We're enhancing the dopamine that's already in the brain. We're either blocking reuptake or causing essentially secretion of the dopamine in the brain. And so that dopamine has to be coming from somewhere. And the two main dopaminergic nuclei in the brain that we've thought about historically are the ventral tegmental area and the substantia nigra. The substantia nigra has been studied heavily in the context of movement disorders, particularly Parkinson's disease. And it's thought that when these nigrostriatal neurons are lost, the patients develop these movement disorders and Parkinson's. Whereas the ventral tegmental area and its projections have been studied heavily in the context of reward, addiction, and things like this. That's sort of the dopamine high pathway, if you will. And it hasn't really been clear where the arousal-promoting dopamine in the brain has come from. In fact, the role of dopamine in arousal has really not been well defined. And one of the main reasons is that the people who study arousal are predominantly sleep neurobiologists. And in the 80s, there were several studies that found that unlike other monoaminergic arousal-promoting neurons in the brain, the dopamine neurons in the ventral tegmental area and the substantia nigra kind of look the same whether you're asleep or awake. They have average firing rates that really don't change over sleep-wake cycles. This is in contrast to sites like the locus ceruleus, which is on when you're awake, and it's essentially quiescent when you're asleep. And those are the kind of sleep switches that people like to see. And so over time, I think people just kind of lost interest in dopamine and thought that, well, it seems to be related to arousal, but we don't really know where it's coming from. It wasn't really clear, and I think these kind of findings made people kind of lose interest in studying dopamine. There was a paper by Cliff Saper's group that was published in 2006. It was a CFOS study that found that there's a cluster of dopamine neurons in the ventral periaqueductal gray area that are also wake active. So they express CFOS more in the awake state than the sleep state. As far as I know, there haven't been any follow-up studies from that, and so this group of neurons hasn't really been well defined. But when you look at review papers regarding dopamine and arousal, this is usually what's cited, the ventral periaqueductal gray, based on this one study. But we wanted to ask the question, well, the two main dopamine sources in the brain are the VTA and the substantia nigra. So if we were to stimulate release of dopamine from those areas, can we induce the same kind of reanimation that I just showed you earlier that we get with dextroamphetamine? So this is the experimental paradigm. This is a coronal section of a rat brain. As you can see, the substantia nigra and the ventral tegmental area, sort of unfortunately for us, are kind of right next to each other. And they're pretty deep, so it's kind of hard to hit the right one. But our approach was to use electrical stimulation, so to put an electrical stimulation electrode into these parts of the brain and use electrical stimulation to have these dopamine neurons in these sites release dopamine and see, can we induce this arousal response during general anesthesia by stimulating dopamine release. So this is one of our rats now under propofol anesthesia. As you can see, he's not in one of those sealed chambers. You can see him breathing right here. I want you to kind of focus on that. He has a tail vein IV. He's on a warming pad, so he's kept normothermic. He's been unconscious on his back for at least 20 minutes now. He has a microstimulation electrode aimed at the ventral tegmental area. And again, I want you to watch him breathing here as the stimulation of the ventral tegmental area occurs. You see that? You see how he started to breathe really fast all of a sudden? So there's this connection between the arousal response and the breathing. And as you can see, we haven't touched the propofol. He's been out cold for 20 minutes. And even under propofol anesthesia, just by stimulating the ventral tegmental area in the brain, we were able to wake this rat up, and he's ready to climb off the table. And so we think that this is pretty good evidence that dopamine release from the ventral tegmental area is involved in arousal and is probably driving the reanimation that we saw with methylphenidate and dextroamphetamine. We did this study and looked at isoflurane and propofol, and we did histological confirmation of where the electrodes were. And what we found was that if the electrode was in the VTA and the animal was under isoflurane or propofol anesthesia, they all woke up. But if it was in the substantia nigra, none of them woke up. And we have 5 out of 5 VTA animals, and essentially 0 out of 5 substantia nigra animals had arousal. And I said this is under review. Actually, we just found out today that this manuscript was accepted by anesthesiology, so you'll hopefully be seeing that in the next few months. So the problem with electrical stimulation, and this is one of the criticisms that we got on this paper, is that it isn't specific for specific neuronal subtypes. And so when you do electrical stimulation of the ventral tegmental area, you're not only stimulating dopamine neurons, there are also non-dopamine neurons there in the VTA, and they're being stimulated too. So you can't really prove that this is a dopaminergic effect just by doing electrical stimulation of the VTA. So we took yet another approach to really try to clearly define this pathway, and we've been using optogenetics, which is kind of a new technique that's been available in the past few years that allows you to specifically target certain neurons within a given neuroanatomical area. And so it allows us to activate just dopamine neurons and not the non-dopamine neurons. So how do we do that? So optogenetics is, the basis of it is really these light-sensitive ion channels called rhodopsins. So they belong to a family of ion channel proteins called opsins, which are actually the chemical basis of animal vision. So this is how we see. This is how our eyes respond to light. These ion channels have a form of vitamin A called retinal incorporated into the ion channel. And when a certain wavelength of light hits that ion channel, the retinal flips from an all-trans form to a 13-cis form. And that conformational change that's driven by light allows this ion channel to open and allows sodium ions to pass through and essentially depolarizes the cell. But we can, by specifically introducing this channel only into the neurons that we want it to be in, we can target specific neuronal populations. And so the way we do that is with a genetic strategy. This is sort of like a real kind of basic slide that explains how we approach this. So we start with a genetic construct that encodes channel rhodopsin, the light-sensitive ion channel. It's flanked by LOX P sites, and so when we take this genetic construct and put it into a virus and inject it into the ventral tegmental area, it will transfect all of this, or hopefully most of the cells, into the area it was injected to. However, what we do is we inject it into a genetically modified mouse that expresses the enzyme Cre recombinase only in cells that express the dopamine transporter. So Cre recombinase is expressed, the expression of Cre recombinase is driven by the dopamine transporter, essentially. Only dopamine cells will express Cre recombinase. And Cre recombinase is required to express this channel rhodopsin. So this is the genetic strategy we use. We transfect all the cells in that area, but only the cells that express the dopamine transporter are actually going to express this light-sensitive ion channel. We then implant that part of the brain with an optrode or fiber optic cable with a laser on it that allows us to shine a specific wavelength of light into that part of the brain. And so this is now one of our genetically modified mice